Our speaker now is Dr. Chris McKay from NASA Ames Research Center to tell us how to go to Mars and beyond.
Thanks. What I'm going to talk to you about is not so much how to go to Mars. We heard about that in the previous session. I'm going to talk about why to go to Mars. And in a sense, what I'm going to say is sort of a introduction to the panel that's going to come up next, where Rick and I talk about the role of NASA and the role of the private sector in commerce and what can each of these sectors do. And I will argue at that panel that what NASA can do is if it focuses on a long-term goal of Mars, then it's doing something it can do, and that has implications for other things it ought to do in the nearer term. So that's why I want to talk about why Mars. So my talk is This Martian Life. And I'm not Ira Glass, but I play him on TV. 3 short stories. So story 1 is second Genesis. We know life started on Earth, or we know life is present on Earth. We assume it started here. Fundamental science question we have about Mars is, did it start there? Was there a second Genesis? And I emphasize a second Genesis, not just life on Mars. And I'll explain why in a minute. So what we're searching for, the big prize, is life, but not as we know it. Here, of course, is Spock and Kirk in Devil in the Dark, episode 26, the original series. You all remember it, of course, where Spock uses his tricorder not just to detect life, but to detect silicon-based life. Quite a remarkable tool. We need his tricorder. And of course, then they shoot it. But it gets to the point that what we're searching for is life, not as we know it. That's what we're looking for on Mars. And we know what we mean when we say life not as we know it now. We know what an alien is. When I was a kid, way, way, way back, an alien was a creature from another planet. So if you met someone, say, are you from another planet? If they said yes, they were an alien. Okay? That was the operational definition of an alien. That is no longer the case. Now the operational definition of an alien is an organism that is not on the Earth tree of life, regardless of what planet they come from. We have learned over the last 20 years that all life on Earth maps into one biochemical and genetic system. That is the life forms from Earth. And an alien, shown here, is a picture of aliens from the secret NASA alien storage locker, is an organism that is not on our tree of life. And in fact, people have suggested that maybe there's aliens here on Earth, organisms, microorganisms that we have not discovered that are not on this tree. Or if we go to Mars, we may find organisms that are on this tree, even though they're on another planet. Then they're still not aliens, even though they live on Mars. It indicates that Earth and Mars have a common ancestor. So where are we going to search for evidence of life? Well, there's lots of worlds in our solar system where we could look. Other worlds with liquids, Mars, Europa, Enceladus, and even Titan. Mars is the closest, and it's also unique in that of all these worlds of astrobiological interest, it's the only one that also is of interest from the point of view of human exploration. That's what makes Mars unique. Not that it's the best place in the solar system to search for evidence of life, but that it's the only place in the solar system where the scientific interest in the search for evidence of life connects with the human interest in human exploration and long-term activities on Mars. So why Mars? And from a science point of view, evidence of past liquid water, the presence of an atmosphere of CO2 and nitrogen, and the cold, dry state, which gives the potential for storing evidence of life. We have a good chance of finding it there. Why isn't Mars habitable today? The short answer is it's too small. Size matters. The problem with Mars is not that it's too far from the sun, but that it's too small. It is shown here to scale with the Earth. Mars is 1/10 the mass of the Earth, and as a result of that, it has no plate tectonics, less gravity, obviously, and no magnetic field. All 3 of those effects, no plate tectonics, less gravity, and no magnetic field, cause Mars to have lost its atmosphere and get cold and dry. All 3 of them. We're not sure of the relative importance of these effects. I've ordered them in the way I think they're important. Different researchers would order them in different ways. But those are the three things that come from being too small. We think a planet has to be this big, half the size of the Earth to be habitable. Mars is 1/10 the size, the mass of the Earth. Okay, back to the search for a second evidence of life. The reason we now worry that if we go to Mars and find life, and it is not a second Genesis, it is part of our same tree of life, is because we now know that rocks have been knocked off Mars, flown through space, and landed on Earth. We have in our meteorite collection some 20 or so rocks that have come from Mars. We know they come from Mars because on the bottom there's a little plaque, made in Mars. No, not really. But just as good. We know they come from Mars because inside these rocks there's little air bubbles of gas, and the gas has the same composition as the Martian atmosphere as measured by Viking. So we have rocks that have came from Mars, and we know that they could have transferred organisms from Mars to Earth. And we assume that the reverse process could also happen. So it's no longer a foregone conclusion that life on Mars would represent a separate origin than life on Earth. They are not necessarily aliens. What we do know about Mars from an astrobiology point of view that makes it important, as I said before, is water, presence of water. And as I was saying, searching for fossils is not enough. Searching for life is not enough. We want to search for a second Genesis. To find that, we need an actual organism because we want to map it on this tree. We need to look at its genetic material and see if it's DNA. And if it is, how does it compare to the DNA in all life forms on Earth? We need the guts of the bug, not just a fossil remnant of it. And that also makes it more interesting in terms of the potential knowledge that we might gain about biospheres, about biology, about microbiology, about the tree of life, about medicine, about agriculture, about all the technologies and quality of life issues that trace back to biology, which are just about all of them. Having another example of biology could put us in a much better position than we are now. Where to go look for it? Well, if I was to pick a spot right now, where is our best chance on Mars of finding evidence of Martian life and the possible preserved biological remains of Martian organisms? It would be in the ancient southern highlands. This is a map of Mars, north, south, showing the heavily cratered regions, showing the ice in the polar regions, and showing the magnetic features discovered by Odyssey. These magnetic features trace back to the earliest time in Martian history. They are arguably the oldest thing we see on Mars. They predate the giant impacts Hellas and Argyre, which erased them in the vicinity, and they predate the north-south dichotomy. So material that still has these magnetic features has been relatively undisturbed for 4 billion years. Cold, undisturbed, ice-rich, drill there, we might find frozen, preserved Martian organisms. Our second Genesis. That's where we'd find it. It's a big job to drill there, but that's where we want to go. Okay, story 2. Story 1 was second Genesis of life. Story 2, a rose. A rose is a rose. A rose on any other world. And this is the story of going out, sending life from Earth beyond. Whenever we draw pictures of Mars bases or lunar bases, there's always a greenhouse. It may be poorly engineered, but it's always stocked with plants. We're going to take plants. We know we're going to stay when we take our plants. If you came to this conference from somewhere else, you probably didn't bring your plants. But if you were moving to California, you would bring your plants. It's an operational test. Are you going to stay? If you're moving to stay, you take your plants. So we got to send our plants. So let's focus on sending our plants. So one of the big pushes I think we need to make with respect to Mars exploration is sending plants. Now, we can do it. Here's an artist's conception. Schrock, a space artist. This is not a technical engineering design for what the plant growth module might look like. It's more representative. But the point is, there's never been a flower on Mars, but we will learn to grow them. Rogers Lasny's wonderful short story, A Rose for Ecclesiastes. And we'll learn to grow them for lots of reasons. It'll be an organism level test of biohazard, environmental effects, Martian gravity. It'll be a technical base for greenhouses, technological and programmatic basis for greenhouses. It's just a cool thing to do, a wonderful opportunity for public involvement. It will be highly symbolic as the first organism to live, grow, and die in another world. It'll help diffuse back contamination issues. It'll be an important biological precursor to human exploration. If this little plant can live, well, then maybe we can live too. And finally, and importantly, it can be done in a way consistent with current planetary protection rules. Plants can be grown without any associated bacteria. You take a rose seed or a tulip bulb and make it completely free of any associated bacteria. And we ought to do it on the moon first. It has the same partial gravity and radiation issues, and it's a lot less stringent in a planetary protection point of view. Mars is planetary protection category 4. The moon is planetary protection category 1, which means it's like Nevada. You can do anything you want there. Leave it up. So we can do it on the moon first. So the mission concept is a lunar plant growth module as a precursor to Mars, as an important step toward human exploration of greenhouses. So here's the Mars, and here's, you might think all I did was take the picture and turn the red color gray to get it to the moon, and you're right, that is all we did. But the point is still a powerful one. So our motto here is fly me to the moon. So now I want to go to story 3. Story 3 takes the story of life to Mars even further, and that is making Mars habitable for life, long-term habitability. We know that Mars is a planet with a past, a past that was wet. Here is a world that was wet. It was a water world. At one time, we had 2 blue marbles in our solar system. Somewhere along the way, we lost one of our marbles. Why? Well, we think we know how it became the cold world it is today, but we also think it could be brought back. How do you bring Mars back? Well, the fundamental challenge here is to warm up the planet. We know how to warm up planets. We're doing it right here on Earth. We are practiced in that technology. And in fact, you could do the same thing on Mars, and you can imagine today and in the future, you notice the difference? Plants. Keeps coming back to plants. When you look at the Earth, it's a planet of plants. You fly over in an airplane, you look down, what do you see? Plants. Animals are small, furry things that run around the edges. The world is dominated by plants. The biosphere is dominated by plants. As I said, we can make Mars warm the way we're making Earth warm with super greenhouse gases. This is just a short diagram of the physics of how they work. They plug up the hole in the greenhouse blanket. We've learned the hard way on Earth not to use chlorine and bromine. Chlorine and bromine destroy ozone. We want to have ozone on Mars. We want to have ozone on Earth. But there's plenty of greenhouse gases that don't involve chlorine or bromine. And this is a calculation by a former grad student, now a postdoc, of the warming on Mars as a function of these different gases per fluorocarbons. And you could warm up Mars appreciably with parts per million of these gases. So this slide is, in a sense, the bottom line. How long would it take? And we can approach the problem essentially as a simple physics problem. Here's Mars today. You think of that as the before state. And here is Mars warmed up. That's the after state. It represents a change in energy, delta E. The only plausible energy source for that energy is the photons from the sun that are incident on Mars. If you calculate how long that would take and put in a reasonable efficiency for warming up planets with a greenhouse effect of 10%, it only takes 100 years of sunlight at 10% efficiency to warm up Mars. So this is not something that is thousands of years in the making or millions of years in the making. That's to warm Mars. On the other hand, to make it oxygen rich, using the efficiency of Earth's biosphere, which is 1/100%, it takes 100,000 years to make oxygen. So we can easily imagine with technologies we understand and have demonstrated that we could warm up Mars. We cannot imagine how we can make it breathable in the sense of Earth's atmosphere. Something, not everything one might like, but something. Why to do it? the fundamental reason, I think, that we would do this traces back to a statement on Feynman's chalkboard the day he left. What I cannot create, I do not understand. What are we trying to understand? This is what we're trying to understand. We're trying to understand the Earth. So I think an important motivation for investigating Mars, investigating life on Mars, investigating the possibility of bringing Mars back to life is the fact that we humans, whether we want to or not, must assume management responsibilities for our planet. There's just too many of us not to assume management responsibilities. It would be a dereliction of duty not to assume that we are responsible and must take it. And we have to learn how to do that, and one of the ways of learning how to understand something is investigating how you would create it. And that is the slide that tells me I'm at the end of the formal presentation, and we can go to questions and answers specific to this before we go to the panel. Question here, Al?
How do you send people to Mars without contaminating, biologically contaminating wherever they go? Or can you?
You can, you do. Let me just answer that question by pulling up some other slides. Can I just... put on my glasses. Sorry, Al, but it's a really good question. And I
figure if anybody can answer it, you can.
yeah, and I do have an answer for it, and I just have to find the answer in my computer.
Your first part of the answer is you can or you can't.
You can. You can send, there is, well, it's not just a question. Let me put up Al's question. Here's Al's question. Don't send humans because they will contaminate Mars before we have a chance to determine if it has life and its potential for life. And it's a very good question. And it's a question that, at least within NASA, we don't ask ourselves, partly because we have two dysfunctional Mars programs, one that focuses only on science and one that focuses only on humans, and they don't talk to each other, and they're bureaucratically separated from each other. They get funding streams, so they view themselves in competition. So right now, NASA has two Mars programs that compete with each other. One rooted in the old vision for space exploration and sending humans to the moon and on to Mars, and one rooted in the Mars exploration program with rovers. And they are not coordinated, and this is one of the places where they must coordinate. And this question of how can you, don't send humans, right? But I want to answer this question by telling you a true story. And that is on July 4th, 1997, bacteria from the planet Earth first landed on Mars. They came unarmed and peacefully. They were, in fact, all asleep. Unfortunately, most died a horrible death by the Martian UV rays. However, there were some survivors. They remain hidden in their spacecraft, dormant, waiting for better conditions. Eventually, in about 1,000 years, they also will be killed by accumulated galactic cosmic rays. So the point of this story, true story, is that we have already invaded Mars. We've contaminated Mars. Now you may say, well, this is the end, that's it. Or you could say this is the beginning. And that story, of course, is Pathfinder. Pathfinder was the first spacecraft to go to Mars, officially not sterilized. Remember, Viking was sterilized, both Vikings, and all the spacecraft that were launched in the Viking era were officially sterilized, just as Viking was officially sterilized. After Viking, COSPAR, that sets the rules, decided that the surface of Mars was unsuitable for growth from life from Earth, and therefore we didn't need to sterilize spacecraft. So we have already contaminated Mars, is an important point. Spirit and Rover each had 100,000 bacteria on them. That sounds like a lot of bacteria, but that's pretty clean. It's cleaner than the dishes in my kitchen. Right? They were clean, but that's still a lot of bacteria. Phoenix was not sterilized except its arm. Its arm was sterilized because it went down and dug into the ice. Right? We know where all these pieces are. Any bacteria that were released from the heat from the spacecraft are dead by the UV light. That was part of the story. They're killed by the Martian UV. Mars has an incredible biocidal UV radiation. The Martian environment is not suitable for any growth of life on Earth. Contamination only survives when it's shielded. So anything inside the spacecraft is still alive, it's still dormant, it's still viable. It's not growing, but it's viable. Anything released is killed instantly. So in a sense, it's contamination of a different type than we're used to when we think of contamination on Earth. And I want to illustrate this difference because it's a very important difference, and eventually I will answer your question, Al. This seems like a long-winded answer, but this is all important background to understanding the answer to your question. Imagine you had rabbits, 2 sets of rabbits, and you took one set to Australia, continent that starts with A. You're going through the alphabet. You took another set to Antarctica, another continent that starts with A, and you released them. And then 10 years later, you decided you didn't want to do that. It was a bad idea, and you're going to decontaminate them. Well, in Australia, that decontamination would be exponentially hard because, in fact, that experiment was done. And the Australians are struggling mightily to get rid of the rabbits. Right? And it is exponentially difficult, essentially impossible. In Antarctica, you would find 2 dead rabbits not far from where you left them. The reason is that Antarctica is not a place where rabbits can grow. It would still be expensive to go get those dead rabbits, right? But it would be linearly expensive. The cost of decontamination is linear with the cost of the contamination itself. And I'm going to call this biologically reversible contamination. This is biologically irreversible contamination. This is biologically reversible. It is not a adjective that describes the size of the contamination. It's an adjective that describes how difficult it is to undo it compared to the cost of doing it. Is it exponentially hard or linearly hard? On Earth, we always think of biological contamination as exponentially hard because most environments on Earth are suitable for life. Mars is not. Let's go to biologically reversible exploration. The idea is to change the basis of planetary detection from preserving science to biological reversibility. It doesn't have much change for current robotic missions. You can still send them contaminated, but you keep track of where everyone is. And if sometime in the future you decide you want to hit the Control-Z button, you want to undo the contamination, you can still do that. And so I'm arguing that the robotic and human exploration of Mars should be done in a way that is biologically reversible. We must be able to undo our contamination of Mars if we discover a second genesis of life. Now, here's an example of something you wouldn't do. This is, you're on Mars, you're commanding your spacecraft, and it says, are you sure you want to drill into this subsurface Martian aquifer? You cannot undo this action. Right? Isn't it wonderful when your computer warns you? We need to extend that metaphor into space. Certain things you would not do, like drill deeply into an underwater aquifer, because if you contaminated that, that would spread exponentially. And to go to your question, this is reversible. This is contamination, but it's reversible. Everything you're doing is left on the surface. These humans are venting organisms from the back of their life support system. Those organisms are being killed by the UV. The organisms inside the containers are still alive. They're there. It's reversible, and it would be costly to clean it up. But the cost is linear, not exponential. So that's my long-winded answer to your question of how do you rationalize a search for life, a search for second genesis, with humans, which are arguably the best tool to do that search, and yet they're a dirty tool. The answer is you don't just give up, you work that tool in a careful way so that you don't do anything that's irreversible. Rex?
When you're talking, Chris, about looking for these new life forms, which may or may not be considered aliens after you map them into our known tree, you're always going to have little. When you're looking for the new life forms, you want to map them into our existing known tree. You always have this problem, won't you, where we never know for sure if that current tree we think is true is the complete tree. So it'll take a little more work, even if you have a life form, to even figure out whether it really maps into the tree, because we might have the tree wrong, right?
Yeah, let me see if I can find the tree in this slide. Yep, there it is. Rex raised a good point. which is it's hard to know what's really not on your tree when you only have one tree. It's hard to know the boundaries of the tree when you only have one, and it's hard to know that you've got the complete tree when you've only got one. In some sense, we'll only be able to answer the question when we've got something else to compare to. And I like to explain it with a simple story. You know, imagine you have a library and all the books are in English, right? Now you get a new book. Let's say you get a new book in Spanish. It's clear that that book has the same alphabet, the Latin alphabet, as the English books in your library do. But it's also clear that it's a different language. On the other hand, you could get a book in Chinese. And it's clear that it's not the same alphabet at all. And in fact, it's even a completely different schema for representing information. And you could have 3 books, English, Spanish, and Chinese, and they all could be books on how to build a chair. And it's curious because at the very bottom level, the books are made out of paper. And at the very top level, the books describe how to make a chair. It's the intermediate level where the books differ dramatically. And that's the problem we anticipate finding in other life. At the ecological level, life on Mars is going to be the same as Earth. It uses sunlight, lives in water, takes up CO2. Ecologically, it's got to be the same because it's living on a planet that's got the similar ecological opportunities and challenges. At the bottom, it's going to be the same, carbon and water. And that's why we go to Mars, because that's an environment with carbon and water. Where we're hoping to find differences is in this intermediate complexity, and that is the complexity represented by this tree. But how can I be sure it'll be different, and how different it will be? Will it be like English and Spanish, which is a hard case to pull apart, or will it be like English and Chinese, obviously different? I can't say until I actually get them there to look at them. Question here? So at least I didn't need 50 slides to answer your question, just one.
Could you talk to us about the part in blue there, the archaea?
The archaea.
And then I have a question behind it after you.
Yeah, okay, thanks for that question. This is this diagram is the original tree of life as mapped out by Carl Wolfs, who was a biologist at University of Illinois, Urbana, who first pointed out that this is the correct mapping for life on Earth. Brilliant piece of work, and he hasn't got the Nobel Prize for it yet, but if they asked me, I would certainly vote for it. This is RNA, and it shows the RNA mapping. There's other ways to map out the tree, but they're all effectively make the same point. And it makes 2 points. One is that all life can group into these 3 domains: bacteria, eukarya, which plants and animals and most of your friends and your neighbors, unless you live in San Francisco. And then you find archaea and bacteria. Archaea, if you flew into San Francisco airport and you saw the pink ponds out in the bay, those are archaea, extreme halophilic bacteria. Normally people think of archaea as sort of obscure organisms that only live in extreme environments like the saturated salt ponds out in the bay. But in fact, they also live inside our intestines. You have about 1% of your gut bacteria are archaea. It's curious because although they live inside us, they don't cause disease. Only these guys, only the organisms in the red states cause disease. The organisms in the blue states don't cause disease. And we don't know why that is, right? And yet what's curious, makes it even more curious, is that these organisms are more closely related to us than these organisms. Here's the common ancestor. They're more closely related. We have archaea in our gut, and about 10% of the population have methanogens. So you actually produce methane. And in cows, the methane is produced by methanogens. The one ecological niche that archaea have that no other microorganism or eukarya has is methane production. And it's very curious. We have no understanding why do these guys not cause disease? Why is methane production the sole province of archaea? To me, it's the favorite question as the type of question that we'll have a better understanding of if we have more than one tree to compare. It's a question about looking down on trees of life rather than looking inside a single tree of life. And understanding why bacteria cause infection and archaea don't could have some important implications.
And so the question I was going to ask is how can we really be sure that the blue isn't already extraterrestrial?
Oh, we know they share I have another slide, but I'm not even going to search for it. All these organisms on this tree have sequences in their ribosomal RNA that are identical. There's a stretch in the ribosomal, in the 16S ribosomal RNA coding for that basically says, I am from Earth. And it's a conserved sequence all through the tree of life. It's the basis for a tree like this. Right? So we definitely know that these are like books that all have the same words. All the genomes of each of these organisms share major paragraphs even, not just words, but entire paragraphs that are duplicated throughout the genome. So it's definitely a single common origin. There may have been other origins that we don't see. There may have been more than one type of life that we haven't seen yet. There may be aliens among us, but we haven't discovered them yet. Partly because the tools we use to search for life are based on this tree. So the only thing we're going to see is things that are on this tree. When you take a sample to a lab and say, is there life in it? What they do is they amplify DNA with PCR. They're not going to see anything that isn't on this tree because what they're using to detect life is the primers that match to this tree. So it's a tight little circle that we go round and round and round.
Why don't we try to, we've talked about sample return versus going there and investigating there. How would you pro or con for either one?
Right now, I'm for sample return, and I base that on the experience we just had with the Phoenix lander. Phoenix went to Mars, scooped up some dirt, and you remember how hard it was to get the dirt through the screen, okay, because the dirt had salt in it, unexpectedly sticky, couldn't get it through the screen. Eventually, we got it through the screen, and then the instrument, the thermal evolved gas analyzer, effectively didn't work because the dirt had perchlorate in it, which is what they use in solid rocket boosters. It's perfectly good salt, but at high temperature it explodes. And so, which is why it's useful as a solid rocket fuel, but it's not useful in an instrument. Right? So the instrument, and it reacted with the nickel on the walls of the oven, and it was just a big mess. So the net result is we got no data, really, from that instrument. Now, if that sample had been in Earth, there's perchlorate in it. Okay, give it to a grad student, rinse the sample, bring it back. 30 minutes later, we're ready to go. Right? And it's an example of how hard it is to do science when you've got to write down all your moves in advance, and you're not allowed to deviate from them. And if suddenly you come across something unexpected, even if it's trivial, you don't have the flexibility to change it. And so I'm thinking that back in the days when spacecraft were just telescopes, looking, that didn't matter. But now when we're in the mode of science where we're trying to pick up dirt and play with the dirt, we're going to hit unexpected things that are going to stymie in situ experimentation. So it's time to bring the dirt home.
It sounds like, though, you're still saying we need to bring the dirt to the person.
Right.
We could take the person to the dirt,
yes,
or the
exactly,
dirt back.
Exactly, yeah. We need the flexibility that comes with a person in a lab, where you can change what you do, you can change, based on results, you can change the protocol and change the instrumentation. Why don't we take one more question, and then we'll go to the panel, where I think we'll broaden the discussion about how to move forward and the role of NASA and private sector. So maybe one more question, if there is one more question. And then Rick will come up and we'll do the panel.
All right. So you just said there was perchlorate in the sand, and so then it ruined the experiment. But I think it's a little extreme to say you got no data because you got a very outrageous data point that you didn't even expect.
Okay, that was from a different instrument, actually, that didn't get ruined by that perchlorate. So there was two instruments on Phoenix, the wet chemistry lab and the thermal evolved gas analyzer. The wet chemistry lab detected the perchlorate. The thermal evolved gas analyzer basically failed because of the perchlorate, and we wouldn't have even known why it failed if we didn't have the results from the wet chemistry analysis. So it's kind of funny because we work in the Atacama Desert where there's plenty of perchlorate, plenty of salts, and they can do all these. We have issues similar like to this with samples on Earth, but we based on the results, we change the protocol. Never do we go into the field and actually execute the protocol that we thought we were going to execute before we went. Now, maybe it's just because we're dullards and not good at planning, and that's true, but still, the flexibility to be able to change what you're doing based on what you found is a fundamental requirement, really, especially when you're dealing with soils as opposed to telescopes, because then all you do is observe. When we move from observers to manipulators, which is what science needs to do, move from observers to manipulators, we've got to change. It's just not working. Okay, last question here. Sorry. This will be the last question.
So just to follow up, how big a lab, how flexible a lab would you have to send to have that flexibility? How much stuff?
Well, you don't need a lot of stuff. A tricorder, a sink, and a fume hood and I'd be happy. But seriously, in terms of a tricorder, some basic analytical instrumentation like we take on field trips, sort of things like dissolved gas analyzers, like Phoenix, a differential thermal analysis, but the critical thing you need is the ability to process the samples before you do the experiment. So that is not so much a bunch of stuff as it is a capability. As a sink, basically it's a small wet lab. And glassware, source of clean water, and some standard chemicals from a kid's chemical set, so that you can titrate acids and titrate bases and neutralize whatever, change whatever problem you come across, you can come up with a solution and work it. So we're not talking about fancy, heavy equipment, but you're talking about the flexibility of having somebody there to do it. That's the hard part. Okay, I'll turn control back over to the moderator.
All right, now we're just going to have a nice discussion about the road forward between these two smart gentlemen.